Today I will tell you about two of our recent studies on adenoid cystic carcinoma then, which is the main focus of our lab. Next slide, please. So first, to give you a bit of a background to the research, I want to tell you about cells, chromosomes, and genes. And it is estimated that the human body contains 30 trillion cells, which is of course an ungraspable number. And you see a schematic of a cell on top here in this image. And in a cell, there is a nucleus where our genetic material is stored. And this is of course DNA, as you know. And the DNA is structured on 46 different chromosomes, as you see here, and an example of. And the chromosomes comes in pairs, so you have 23 chromosome pairs in a cell nucleus. And if you would take the genetic material of one cell and stretch it out, it would become 2 meters in length. So you understand that it's completely super densely packed into each cell nuclei. And the way it is packed is that it's wrapped around a special type of proteins called histones. And the DNA itself is made up of 4 building blocks called A, T, C, and G. And this is the genetic code that you have heard about. And the sequence of the genetic codes is what makes each individual unique. Along the genetic code, or the DNA, there are also regions termed genes. And humans have 21,000 genes. And it is when these genes are damaged or altered in some way that cancer may develop. And there are special types of genes called cancer genes where this happens. Next slide, please. So one such cancer gene is the MYB gene that has been a focus of our lab for a long time. And back in 2009, we showed that this cancer gene can get activated by a specific mechanism called gene fusion. And this mechanism is specific for adenoid cystic carcinoma, not present in any normal cells. And what is happening here is that there is a translocation of genetic material between chromosomes 6 and 9, resulting in a completely new gene in ACC. And this gene is partly formed by the MYB gene and partly by another gene called NFIB, residing normally on chromosome 9. And MYB is residing on chromosome 6. So subsequent to our studies, other groups have demonstrated that the MYB gene can get activated in ACC by other mechanisms as well. So this gene is really central to ACC. It's an early event in the disease, this activation of the MYB gene, and it's also a potential target for therapy, therefore. There is also another gene called MYB-like1, which is related in structure and function to MYB, which is also activated in a subset of cases, a smaller subset of cases where MYB is not activated. So it can replace MYB in these cases. Next slide, please. So why does MYB activation lead to ACC development? The MYB gene encodes a master transcription factor. And the transcription factor is a gene-protein product that can regulate the activity of other genes in the DNA or in the genome, that it's also called, in the cell. And the master transcription factor can regulate the activity of other transcription factor coding genes. So this means that MYB can control many of the central processes in a cell. It's a very powerful gene in that way. And these processes are, for example, cell proliferation, that is cell growth, cell survival, differentiation. And these processes are central to the development of cancer. So our group together with Arturo Sala's lab, has previously shown that MYB activation leads to a transformation of normal cells into tumor cells, and also that MYB activation drives ACC growth. So this makes the MYB activation a very central part of ACC biology, and also then a potential or very important target for the therapy. Other groups have also shown that MYB can be activated in, for example, certain leukemias and in subset of breast and colon cancers. But the gene fusion that is found in ACC is specific for just ACC. Next slide, please. Although several studies have shown the importance of MYB activation in ACC, we wanted to investigate what is the frequency of these gene fusion events and other rearrangements of the MYB gene in ACC, and what is their clinical significance. So this is what we have done in the first study I want to present today. We try to answer this question. Next slide, please. So in the first study, we made use of a method called fluorescent in situ hybridization, or FISH for short. And this is a method where you can detect the localization of a specific gene on a chromosome or in a cell nucleus. And you make use of patient tissue sections, and you use a specific type of DNA probe, which is a complementary DNA molecule that can bind the gene of interest. You can label these DNA probes with a fluorescent tag, which light up in a special type of microscope. And when you treat the patient tissue section with this probe, it will bind and light up the gene you're looking for. So this means that you can find the localization of a gene on a chromosome or in the cell nucleus. Next slide, please. So the first study was a collaboration of several different centers, both in Europe and in the US. And we used the FISH method to analyze more than 500 cases of ACC. We also validated our findings in more than 1,000 non-ACC salivary gland tumors. And we had clinical follow-up data available for 366 ACC patients. And in this study, we used the FISH method, and we also used another method called immunohistochemistry, where you can detect the protein product of the MYB gene in the tissue samples. It's fine. So these are the results from the FISH experiments, where we are looking for rearrangements of the MYB gene and MYB-like 1 gene in ACC. So that is, yes. And if you look to the panel or the image on the top left here, the blue objects that you see are cell nuclei. And the dots that you see, this is the signal coming from the DNA probes we use with the FISH method. And in this case, we are using probes, 2 probes for the MYB gene, binding different parts of the gene. And since the chromosomes come in pairs, you have 2 MYB genes per cell, or per cell nuclei. So you would expect 2 pairs of dots sitting together. And in this image, where the arrowheads are indicating a separation in one of the pairs, meaning that parts of the MYB gene has moved away or broken up and are located topologically somewhere different in the nucleus. And this is an indication of this gene fusion event, or this translocation of genetic material between chromosomes. So this is a rearrangement called MYB split. And this MYB split was found in 39% of the analyzed cases. It was not found in any of the more than 1,000 other non-ACC salivary gland tumors, making it highly specific for ACC. Another rearrangement, which was the most second common we found, was loss of the 3' part of MYB. So this is shown in the upper right image. And there we have lost one of the red dots. So we're losing parts of the MYB in the cell nuclei. So it's deleted from the DNA, this part of the MYB gene. And this was found in 16% of the cases, as shown in this pie chart up in the upper right here. Another rearrangement which was found in a few percent was MYB gain in the lower left panel. And here you can see that there are 3 pairs of dots per cell nuclei, which means that there is an extra copy of the MYB gene in these cells, also activating MYB in this case then. When it comes to MYB-like-1, we also found split in this gene, as shown in the panel to the lower right, but in much less frequency, and only 7% of the cases have this MYB split. And in total, we found that 62% of the cases had some kind of MYB rearrangement, and 16% had some kind of MYB-like-1 rearrangement. So all in all, 78% of the cases had either MYB or MYB-like-1 rearrangements. Next slide, please. We also used this method called immunohistochemistry to detect the MYB protein in patient tissue sections. And here we make use of antibodies that are detected against MYB. And we analyzed close to 300 cases with this method. And in 93% of the cases, we saw an activation of MYB or an overexpression of the MYB protein, which means that there is an activation of the gene. And it did not matter which type of MYB rearrangement the case had, we still saw this activation. There were also many cases that had activation or protein expression of MYB, but did not have an rearrangement by the FISH method, which means that there are alternative mechanisms of MYB activation that are not related to the gene rearrangements we can see with the FISH method. And this has also been shown by several studies, this alternative mechanism of activation. There are also 3 different morphological subtypes of ACC, and we did not see a difference between these in the terms of the MYB protein expression. So all of them expressed MYB, the MYB protein. There were also some cases that were, for example, positive for MYB-like-1 split, but not showing any MYB gene rearrangement using the FISH method. And these still expressed the MYB protein or using the immunohistochemistry. So we believe that the antibody that we use to detect the MYB protein may also detect the MYB-like-1 protein because it's highly related in structure. Next slide, please. So to further investigate the clinical significance of MYB and MYB-like-1 gene rearrangements, we performed survival analysis in cases having these rearrangements or lacking these rearrangements. And in panel A, we see the results from the analysis where we have compared MYB rearranged cases with cases that don't have detectable MYB rearrangements. And we could not see a significant difference in terms of the overall survival of the patients in this analysis. The same was true in panel B for MYB-like-1. We did not see a significant difference here either. Then we wanted to investigate the 2 most common MYB gene rearrangements, MYB split and loss of the 3' part of MYB, if there was any association with survival in these, with these rearrangements. And in panel C, we have investigated MYB split. Again, we did not see a difference in terms of survival. But however, in panel D, you can see a comparison between cases that has loss of the 3' part that is losing one of these dots in the cell nuclei, compared to cases that lack this rearrangement. And you can see that loss of the 3' part of MYB was significantly associated with shorter overall survival of the patients. We could also see that in panel E, loss of the 3' part of MYB was found in tumors of all different grades, but were more common in high-grade tumors. We did not find an association with patient age at diagnosis and this rearrangement, neither the sex of the patient or if there were any perineural invasion present in the case. Next slide, please. So to further validate loss of the 3' part of MYB gene rearrangement as a prognostic biomarker, we performed multivariate analysis. And in this analysis, we could show that loss of the 3' part of MYB together with age and grade 3 were significantly independent prognostic biomarkers. Yes, next slide, please. So to summarize this part, we investigated MYB and MYB-like1 rearrangement using the FISH method, and we found that they occur in 78% of the ACCs, and we found overexpression or activation of the MYB protein in 93% of our analyzed cases. This makes MYB and MYB-like1 key diagnostic biomarkers for ACC and also potential targets for therapy. We could further show that loss of the 3' part is more common in the high-grade tumors, and it was also an independent prognostic biomarker for survival in a multivariate analysis. So the results from our study indicate that there are genes with tumor suppressive functions that are lost when this 3' part of MYB is deleted from the cell nucleus. Next slide, please. So in the second study, which was recently published, we have investigated global genomic alterations in ACC, and in particular, this type of alterations called copy number alterations. Next slide, please. And copy number alterations, or CNAs, is a alteration of the amount of genetic material present in each cell nuclei. So you can have a gain of genetic material, so you have more DNA than you would commonly expect, or you can have a loss of material, for example, so you have less genetic material. And this can be whole chromosomes that are lost or gained, or you can have parts of the chromosomes that are gained or lost. And in the upper panel, the case for normal cells is shown, where you have no net gain or loss of genetic material. So this is a normal cell. In the lower panel, I have taken an example of an aggressive breast cancer, where you can see several gains here in red and several losses in green. And this is parts of different chromosomes that are either gained or lost. You have a change in the normal amount of the genetic material here. Next slide, please. In this study, we have used a method called array CGH analysis to detect these copy number alterations in ACC. And we performed the analysis of 100 ACCs. We also used the FISH method again to validate our findings on close to 500 ACCs. And we performed global gene expression analysis of 30 ACCs. And this is a way to simultaneously measure the activity of all 21,000 human genes. Then we also did some experimental confirmation or validation in cultured cells from 2 ACC cases. And we had follow-up data for several hundreds of ACC patients. Next slide. So this is an overview of the results for copy number alterations in ACC using the array CGH method. And we could see generally here, if you focus your attention to panel B, we could see that generally ACC has a stable genome, and in this case 66% of the cases have 5 or less copy number alterations in the entire genome. Copy number alterations were more common in high-grade tumors, grade 3, if you look in panel A, compared to tumors of grades 1 and 2. And we also identified several recurrent gains and losses of genetic material in ACC. And we made use of a software called Gistic, which identified 4 focal regions of chromosomal deletion in ACC. And these were in the chromosomal regions 1p36, 6q, 9p, and 12q. And when we performed survival analysis of these deletions, as seen in panel C, we could see that the 1p36 deletion and the 6q deletion were significantly associated with a shorter overall survival of the patients. This suggests that there are genes within these regions that normally has a tumor suppressive function, but are lost in these tumors, which correlates with the shorter overall survival in the patients. Next slide, please. So to investigate potential genes within the 6q region on chromosome 6, we again made use of the Gistic software, and this software identified a minimal common region in 6q that contained the PARK2 gene as the only gene in this region. The PARK2 gene is known to regulate cellular processes such as cell proliferation and programmed cell death, which is also called apoptosis. So in 28% of the cases, we found PARK2 deletions in ACC. And in panel B, we have used a method called quantitative real-time PCR to quantify the activity of the PARK2 gene in ACC cases. And what you can see here is that cases that have a 6q deletion have a significantly lower activity of the PARK2 compared to cases that lack these deletions. So this suggests that the deletions that we show in panel A correlate also with the gene activity, so which goes down then in these cases. Next slide, please. Yes. So to test the hypothesis that the PARK2 gene has tumor suppressive functions in ACC cells, we made use of cultured ACC cells from 2 different cases. One of the cases, the ACC100 cells, shown in panel A, have a 6q deletion. And the other case that we studied, ACC67, has an intact 6q deletion. Region, 6q region. We could grow these cells as small microtumors in the lab, or spheroids. And when we compare the growth rate of these spheroids, we could see that the ACC100 cells having the 6q deletion grew much faster than the other case. And to investigate whether PARK2, the gene PARK2, was mediating this effect, we used a method called RNA interference, by which you can knock down the gene activity experimentally in the cells. So the results from this knockdown experiment is shown in panel B. And we could see a potent knockdown of the PARK2 gene activity here, also at the protein level as shown in the right of panel B. And this experimental manipulation, when we did that on the ACC67 cells, which had an intact 6q region, they started to grow faster. And these results are shown in panel C. So they formed these microtumors to a higher rate when you knock down PARK2, suggesting that the PARK2 normally blocks growth in ACC when it's intact. So PARK2 is also known to be involved in regulation of programmed cell death or apoptosis. So we wanted to test if the 6q deletions affected the apoptosis in the ACC cells. So we treated, in panel D, we treated ACC cells with the chemotherapeutic drug doxorubicin, which is known to induce apoptosis, and we could see that the ACC100 cells having the 6q deletion were less sensitive to the drug, showing less apoptosis. And again, to check if, or to test if the PARC2 gene was responsible for this effect, we knocked down the gene activity as we did in panel B. And in panel E, you can see the results when we have treated the cells simultaneously with doxorubicin and this PARC2 gene knockdown. And the PARC2 gene knockdown caused the cells to be less sensitive to the programmed cell death induced by the drug. So this suggests, or we can show both genomic evidence and experimental evidence now that the PARC2 gene has tumor suppressive function in ACC, and when it's lost, the ACC cells seemingly grow faster and are less sensitive to chemotherapy. Next slide, please. So to study the molecular consequences of 1p36 deletions in ACC, we made use of our global gene expression data in 30 cases. And we focused on chromosome 1. And you can see in panel A, you can see the deletions here as a red, it's like a red dot or like red field there. There you have the copy number alterations in all the 30 cases showing downregulation. And we could see that the gene expression the gene activity of genes within this region was decreased. So we could also confirm this data using a method called gene set enrichment analysis, which is shown in panel B. And further to try to understand which are the biological processes or cellular processes associated with the downregulation of this gene within 1p36, we made use of a method called gene ontology analysis. And this analysis identified apoptosis, again, as a top process here. And in particular, we found that the genes TP73, MFN2, KIF1B, DFFA, and DFFB were significantly downregulated in tumors having 1p36 deletion compared to tumors lacking this deletion. And this data is shown in panel A. So this suggests that downregulation of these 5 genes in ACC having 1p36 deletion makes the cells less responsive to apoptosis. Next slide, please. So to test this experimentally, we again made use of this RNA interference method where we can knock down gene activity in the cultured cells, ACC cells. And we did this for the early pro-apoptotic genes TP73, MFN2, and KIF1B. And the results of this experiment is shown in panel A, where we got a robust knockdown of gene activity using this RNA interference method. And we again treated the cells in conjunction with this with the doxorubicin drug, which causes or induces apoptosis. And we could see that when we knocked down TP73 and KIF1B, we could see that the ACC cells got less sensitive to the drug and less, there was a smaller induction of apoptosis. For the MFN2 gene, the effect was much less or in some cases absent. We also confirmed our results with another drug called vinorelbine, which is also inducing apoptosis and is a chemotherapeutic drug. Next slide, please. So to validate our genomic findings, we again used the FISH method, and in a separate cohort, larger one, we try to investigate at what frequency are these rearrangements present. So we use FISH probes that could detect 1p36 deletion, but also the PARC2 deletions. And we found 1p36 deletions in roughly 14% of the cases, while PARC2 deletion was found in 24%. And we could also see that both of these deletions was associated with a significantly shorter overall survival of the patients, as seen in panels A and B. And simultaneous deletions of both 1p36 and PARC2 was also significantly associated with shorter overall survival compared with cases having one of these deletions or neither of them, as shown in panel C. And the simultaneous deletion of 1p36 and PARC2 were exclusively found in grade 3 tumors. And we could also show that the 1p36 deletion was a significant prognostic biomarker together with age, grade 3 and stages 3 and 4 using multivariate analysis. Next slide, please. So to investigate the potential association between genomic and gene expression profiles in ACC, we used unsupervised hierarchical clustering of our global gene expression data from 30 ACCs with known genomic profiles. And the results of this study is shown in panel A. We found that the 30 ACCs cluster into two separate groups that were distinct from the normal salivary gland samples we used as control, here termed NSG, on the right of panel A. We found that the ACC1 cluster contained all the cases with 1p36 deletions. The PARC2 deletions were found in tumors of both clusters, but were somewhat more common in the ACC1 cluster. And the MYB gene was activated in all tumors except for one, which the MYB-like 1 gene was activated instead. And if you look at the ACC1 cluster there are four tumors that lack the 1p36 deletions. But we found that they instead have a downregulation of the TP73 gene activity, which is found in the 1p36 deletion. So this seems to be a common theme in this group. And we can show that also in panel B, that all of the ACC1 tumors had low TP73 activity compared to the ACC2 group. And importantly, there was a difference in the survival of the patients between these two groups, where the ACC1 patients with the ACC1 tumors had much shorter overall survival. So taken together, we can show that genomic as well as gene expression profiles are significantly associated with the prognosis of ACC. Next slide, please. So to study the biological processes associated with these gene expression profiles, we used again the method called gene set enrichment analysis. And we found that in the ACC1 tumors, they had gene sets that are associated with metastatic and invasive tumors. The ACC1 tumors also had a downregulation of genes involving cell adhesion and extracellular matrix organization. And they further had an upregulation of genes involved in cell proliferation. So this data suggests that the ACC1 tumors, they grow faster than the other tumors, and they're also more prone to invade and metastasize. Next slide, please. So to summarize, we find that 1p36 and PARC2 deletions are prognostic biomarkers in ACC. And we also find that PARC2 is a new tumor suppressor gene in this disease. And when TP73 and KIF1B genes are lost within the 1p36 region, this contributes to a more advanced disease that seems to be more resistant to apoptosis and chemotherapeutic drugs. And deletion of the 1p36 was also an independent prognostic biomarker in multivariate analysis. So finally, the global gene expression profiles that we found also correlates with the survival in ACC. Next slide, please. So I would like to thank the members of the Göran Stenman lab in Gothenburg and also our collaborators in Europe and in the US. I would also like to thank the founding agencies making this research possible. And last but not least, the patients that are generously donating tissue samples for research. This is very important.